Hi, my name is Leah, and today I will be talking to you about determination of gender, genes versus hormones. For the sake of this presentation, when I refer to gender, I will be referring to the biological differences that make males males and females females. Technically, this refers to an individual's sex, whereas gender is usually more related to social roles, but for the purposes of this presentation, this is what I mean when I refer to gender. By analyzing our DNA, we can look into the genetics behind many traits such as hair and eye color, presence or absence of certain diseases like Down syndrome, or even identify the gender of an individual. In order to procreate, a sperm cell needs to fertilize an egg. Each of these cells donates 23 individual chromosomes to the resulting zygote. The egg and sperms themselves are considered haploid because they have a single set of unpaired chromosomes. 22 of these chromosomes are autosomal chromosomes, while one is a sex chromosome. When the egg and sperm unite, the resulting zygote contains 46 chromosomes. One chromosome of each matched pair comes from the mother and the other from the father. The resulting zygote is considered diploid, meaning diploid because it has duplicated chromosomes. The mother's egg can only contribute an X chromosome, while the father can contribute either an X or a Y chromosome. In the absence of a Y chromosome, a female will develop. Even if there's only one X chromosome inherited, which would be an X and an O, also known as Turner syndrome, a female will still develop. However, 2 X chromosomes are needed in order to have normal female reproductive function. Therefore, a typical female will have 2 X chromosomes. The Y chromosome is essential for the development of the male reproductive organs. Even if a chromosome has extra chromosomes, so for example, an XXY set, the zygote will still develop as a male. However, if a zygote inherits only a Y chromosome, aka a YO, the zygote will be unable to develop and will die because it is missing the essential genes contained on an X chromosome that the Y chromosome lacks. The sex of an early embryo is difficult to determine early in a pregnancy because the reproductive structures have not begun to differentiate. They do so after the 7th week of development. Until this differentiation occurs, the embryonic tissue is considered bipotential because it cannot be identified as male or female quite yet. The bipotential gonad has an outer cortex and an inner medulla. The medulla will develop into the testes under the appropriate developmental signal. However, if the signal is lacking, the cortex will develop into ovarian tissue. The bipotential internal genitalia consists of 2 pairs of accessory ducts. The Wolffian ducts that derive from the embryonic kidney, and the Mullerian ducts. As development continues, only one of these ducts proceeds with development while the other one degenerates. Which duct proceeds and which degenerates depends on gender. The bipotential external genitalia consists of a genital tubercle, urethral folds, urethral groove, and labioscrotal swellings. These structures will all differentiate into the male or female reproductive structures as development continues. But what directs a single-celled zygote to become either male or female? So sex determination depends on the presence or absence of the sex-determining region of the Y chromosome, otherwise known as the SRY gene. In the presence of this gene, the bipotential gonads mentioned earlier develop into testes. In the absence of the SRY gene, and under the direction of multiple female-specific genes, the gonad develops into ovaries. The presence of the SRY gene produces the protein testes determining factor, which binds to DNA and activates additional genes. The products of these direct the development of the gonadal medulla into testes. Once the testes differentiate, they secrete 3 hormones that influence the development of many internal and external genitalia. Testicular Sertoli cells secrete anti-Mullerian hormone, which causes the embryonic Mullerian ducts to regress. The Leydig cells secrete the androgens testosterone and dihydrotestosterone, or DHT. Testosterone converts the Wolffian ducts into male accessory structures such as the epididymis, vas deferens, and seminal vesicle. Later on in development, testosterone also plays a role in the migration of the testes from the abdomen to the scrotum. DHT, on the other hand, is responsible for the differentiation of the external genitalia. Testosterone and DHT are the dominant steroid hormones in males. The female embryos, on the other hand, have no SRY gene, so the cortex of the bipotential gonad develops into ovarian tissue. Without testicular AMH, the Mullerian ducts develop into the upper portion of the vagina, the uterus, and the fallopian tubes. Without testosterone, the Wolffian ducts degenerate, and without DHT, the external genitalia takes on female characteristics. So in summary, the genetic determination of gender results from the presence of a Y chromosome or just two X chromosomes. In contrast, the hormonal gender determination results from the presence or absence of the SRY gene, and the domino effects that result from either its presence or its absence.